Hi, my name is Jen Bomberger. I'm an associate professor at the University of Pittsburgh. I'd like to start by thanking the Cystic Fibrosis Foundation, as well as the session organizers, Lisa and Matt, for inviting me to give this talk. Today, I'm going to give a talk on the year in review, the most influential papers in CF microbiology focused on the basic sciences from the year 2020. I want to make a note at the beginning of the talk that I'm going to be following the chat throughout the conference, so if you'd like to discuss or leave comments on some of these papers, I'm happy to engage there since we can't in person this year. My disclosures are I serve on the Scientific Advisory Board for Emily's Entourage. I also receive grant funding from the CF Foundation, Gilead, and Vertex. So today I'd like to take you on a tour through the literature from the past year, so 2020 papers in CF Basic Science Microbiology. So these are some of my favorite papers, as well as I've surveyed some CF microbiologists and tried to select the favorites from the group and present those here today. So I'm going to take the selected papers and I broke them into categories to help with summarizing some conclusions. My topic categories are microbial response to environmental triggers in CF, microbe-microbe interactions, host pathogen interactions, and new and improved models and tools that I think are going to be important for CF microbiology moving forward. So we're going to start with environmental triggers in the CF respiratory tract and the microbial response to them. So the first trigger that we have to think about is antimicrobials and cystic fibrosis. And this is a paper, a really nice study by Vaughn Cooper and his team at my home university, University of Pittsburgh. And so Vaughn's team looked at the evolution of antimicrobial resistance to tobramycin, which is a frontline antimicrobial used in CF. And interesting in this study, they looked at two different bacterial species, Acinetobacter baumannii, as well as Pseudomonas aeruginosa. The way they did their evolution studies, they looked at evolution of antimicrobial resistance to a dose escalation of tobramycin over 12 days. And they looked at both evolution of antimicrobial resistance in a planktonic lifestyle of the bacteria, as well as a biofilm lifestyle. When they did these studies, they actually observed that parallel evolution of tobramycin resistance was observed over the study, both at the species and the environment level. This was very interesting. They also then took a population sequencing approach and saw that they also observed gene level parallelism, mutations in both the genes FUSA1 as well as PTSP. So FUSA1, they observed these mutations in regardless of lifestyle, so planktonic and biofilm, as well as across two different species of bacteria. They saw for PTSP mutations, these were observed in both lifestyles for Acinetobacter as well as the planktonic population for Pseudomonas. So the rest of the study, they focused on the FUSA1 mutations and observed that if they looked in the literature for other microbes exposed to aminoglycosides, they also observed FUSA1 mutations. And this was true for E. coli, Staph aureus, and Salmonella enterica. If they then looked at CF clinical isolates and the genomes that have been published for them, very frequently we see FUSA1 mutations. And this makes sense in CF with the frequency of tobramycin cycling in the clinical care of CF patients. And so this suggests that the way bacteria evolve to antimicrobials may span both the lifestyle of the bacteria as well as bacterial species, which I thought was interesting. The second environmental trigger that I wanted to talk about was microoxia, and this was addressed in a very elegant study by Deb Hogan's team at Dartmouth in the past year. And so Deb's paper started with looking at the LASR gene and LASR null strains. And so LASR we know encodes a quorum sensing regulator, and this is these deletion mutations in LASR are very commonly seen in cystic fibrosis clinical isolates. What we haven't known over time, though, is why did these strains, these LASR null strains, seem to be selected for in the CF respiratory tract? And Deb's work from the past year really, I think, has shed some light on this. And so what Deb has shown is that LASR null strains are more fit in microoxic conditions, and this is dependent on the transcription factor A and R. And so if you look in the blue dots here for wild-type Pseudomonas as compared to the purple squares, we see the purple squares are more fit in low oxygen conditions, and this is the LASR mutant. If we take this LASR mutant that is more fit and use that as a background and delete ANR, the transcription factor, we see that this increased fitness is dependent on ANR, and Deb's team can complement this back by complementing ANR. And so what Deb has shown in these studies is that Pseudomonas success in chronic infections is in part due to its propensity to lose LASR functionality, and that this, the reason this is selected for in the CF respiratory tract is this gives increased fitness in a microoxic condition, which we know are common in the CF respiratory tract. So what these studies have shown us is new mechanisms underlying the ability of Pseudomonas to persist and compete even in a stressful environment like the host respiratory tract in response to antimicrobials. And it's also shown that there's conserved mechanisms potentially existing for evolving antimicrobial resistance, and this can cross both species and lifestyle. So I thought that was a very interesting observation. So for microbe interactions, the first paper I'd like to highlight is from George O'Toole's team at Dartmouth, looking at Staph aureus and Pseudomonas aeruginosa interactions. And so we know Staph aureus and Pseudomonas aeruginosa are frequently co-isolated from a number of infection sites in a number of different disease models, including the cystic fibrosis respiratory tract. Recent clinical studies in CF have also shown that co-infection with Pseudomonas and Staph has also been shown to produce worse outcomes in patients than if they're infected with either organism alone. And so a lot of interest in the field has gone into the past few years looking at interactions between Staph and Pseudomonas in terms of pathogenesis in CF. I also need to note George's team has published a suite of papers, excellent papers in the past few years, looking at how microbe interactions they're now uncovering are really important in changing antimicrobial susceptibilities of microbes. And so this is another paper in this suite of studies that George's team has done. And so in this case, they're looking at Staph aureus biofilms and treatment with norfloxacin, the antimicrobial. And so here they see that Staph aureus biofilms alone treated with norfloxacin are only minimally susceptible to norfloxacin. But if you co-incubate these Staph aureus biofilms with Pseudomonas or Pseudomonas supernatans, we see a dramatic reduction in Staph aureus biofilm growth, an increase in the susceptibility of Staph aureus to norfloxacin. And so George's team uncovered the mechanism for this and identified several Pseudomonas exoproducts that are actually increasing this susceptibility of Staph aureus biofilms to norfloxacin. And this is through an increase in membrane fluidity and permeability that drop the membrane potential and allow norfloxacin to accumulate intracellularly in Staph. The next paper I'd like to highlight is a paper looking at interactions between Pseudomonas aeruginosa and the Burkholderia cepacia complex in cystic fibrosis. This is an excellent study by Peggy Cotter's team at UNC. And so they start the study by highlighting the fact that Pseudomonas aeruginosa can initiate long-term infections in young CF patients that can persist throughout life. But Burkholderia cepacia complex infections often arise in teenagers and adults, and you often will not see Pseudomonas and Burkholderia in the same patient's respiratory secretions. And so Peggy's team was very interested in the interaction between these two microbes. And so what they observed is that Pseudomonas isolates from teenage and adult CF patients, so older CF patients, but not those from young CF patients, can be outcompeted by the epidemic Burkholderia isolate, and they do this in a type 6 secretion system manner. And so I should first note that the type 6 secretion system is a bacterial secretion system that works like a syringe that basically requires contact dependence between two microbes where they can shoot each other with these syringes and inject toxins in the neighbor and kill them. So it's important to note that both Pseudomonas and Burkholderia can have encode and have these type 6 secretion systems. But what Peggy has uncovered on her team in these studies is that Pseudomonas aeruginosa actually, as it's evolving in the respiratory tract in CF, they evolve loss of function mutations and they lose expression of the type 6 secretion system. And so what they've uncovered is the mechanism for this competition, and the way Burkholderia can outcompete Pseudomonas over time is that Burkholderia continues to express its type 6 secretion system, and when it encounters a Pseudomonas aeruginosa that does not have type 6 secretion system, it's able to kill that bacteria and allows Burkholderia to take over and rise in abundance in the population. And so Peggy's team has also shown that they can take these type 6 secretion system mutants in Pseudomonas, repair those mutations, and they can now compete with Burkholderia again. So I thought this was a fascinating study with two microbes that we don't often study their interactions in CF. And the final study I'd like to highlight in the microbe interactions is looking at Stenotrophomonas multifilia and its interactions with Pseudomonas aeruginosa in a polymicrobial airway infection model and a mouse model. And this was excellent work done by Ed Sword's team at UAB in the past year. So Stenotrophomonas is one of those neatly, very highly antibiotic resistant gram-negatives. It's quite problematic when it's acquired by CF patients. And trying to understand how it's acquired and how it persists, I think, has been important. And there hasn't been as much literature there as and attention as there should be. And so Ed's team did a great study showing that polymicrobial infections with Pseudomonas aeruginosa increase the bacterial burden of Stenotrophomonas in this lung infection model. When they looked at this increase in Stenotrophomonas in the presence of Pseudomonas, they saw that the bacterial load of Stenotrophomonas was directly correlated with the density of Pseudomonas, and it required live Pseudomonas to elicit this increased bacterial load as well as persistence in the mouse infection model. And so interestingly, when co-infected, both of these microbes are able to persist in the mouse lung, which individually they cannot. And they also together induce enhanced pro-inflammatory cytokine secretion, which is detrimental, particularly in the CF respiratory tract. And so I thought this was another microbe-microbe interaction study that was excellent, focused on two microbes in CF that we don't often study their interactions. And so together, I think concluding thoughts from this section is, I think these papers and other papers related that I didn't have time to talk about uncovered microbial-derived products that can alter the ability of antimicrobial agents to kill bacterial biofilms. They've improved our understanding of how interspecies interactions can change antimicrobial susceptibilities in bacterial biofilms and could inform treatment decisions moving forward, as well as be considerations when we're developing new antimicrobial therapies. And also this increased awareness and study of microbe-microbe interactions in the CF respiratory tract, I think, is going to help our understanding in understanding CF pathogenesis. So host pathogen interactions is the next category. And the first paper I'd like to talk about here is looking at the gender gap in CF microbiology. So this is an excellent study done by Brian Harvey's team at the Royal College of Surgeons in Ireland. And so this paper builds on some previous research that CF lung infections display sexual dimorphism in CF, where females with cystic fibrosis develop more serious infections with Burkholderia. They're colonized earlier with Pseudomonas aeruginosa, and this results in a more rapid loss of lung function that correlates with the onset of puberty as well as the menstrual cycle. Previous publications have also shown that estrogen can induce the conversion of Pseudomonas to mucoidy through MUC-A mutations. And so we know there's an interaction with estrogen with Pseudomonas aeruginosa. The new contribution for this paper was that estrogen can induce a number of different virulence mechanisms in Pseudomonas, which is new knowledge. And so I'm showing you here on the right an increase in the twitching motility observed in the blue bars with estrogen treatment. And interestingly, the part I think is really exciting about this paper is the authors show that using selective estrogen receptor modulators, so these are clinically approved therapies, you can actually reduce this enhanced virulence back to baseline. And so they propose this as a potential adjuvant therapy for antimicrobials with Pseudomonas treatment. And so I thought that was fascinating. The next study I'd like to talk about is looking at Pseudomonas and its response to host metabolism. This is a great study done by Alice Prince's team at Columbia in the past year. And so in this study, Alice Prince and her team show that Pseudomonas alters its metabolic and its immunostimulatory properties in response to a macrophage metabolite called Itaconate. And so in this study, Alice's team shows that Itaconate is able to induce bacterial membrane stress, and this results in a downregulation of LPS, as well as an upregulation of extracellular polysaccharides, which drives enhanced biofilm formation. They also go on to show that if you look at CF clinical isolates, these Itaconate-adapted Pseudomonas are able to accumulate mutations that favor Itaconate assimilation and biofilm formation. And so if you look on the right-hand side at CFU from Pseudomonas in the lung infection model in mice, in the Itaconate, in the mice that can produce Itaconate by their macrophages, we see an increased bacterial burden as compared to mice that are no longer able to make Itaconate. And this trend only holds for CF clinical isolates. If we look at the lab isolate Pseudomonas or PA01, we do not see this phenotype. And so this is this idea of Itaconate adapted Pseudomonas that have this enhanced Itaconate assimilation and biofilm formation. And really this elegant study really brings to point that we need to consider Pseudomonas's metabolic versatility when we think about designing new therapies and new antimicrobial treatments, which I thought was fascinating. The final paper in the host pathogen interaction section that I'd like to talk about is this excellent study looking at changes in Pseudomonas virulence in response to mucus and mucins. And this was a beautiful study done by Katerina Ribek's group at MIT in the past year. And actually, I should say it's late 2019, but I like this paper so much, I decided to throw it in anyway. And so the authors show in this paper, very interestingly, that exposure to mucus is able to downregulate virulence genes in Pseudomonas aeruginosa that are involved in quorum sensing, siderophore biosynthesis, and toxin secretion, and it's also able to rapidly disintegrate biofilms. And then they went on to show that the actual mechanism here is that the active agent in the mucins that are able to have this downregulation of virulence is actually the O-linked glycans in mucins like MUC5AC. And so here on the right, I'm showing you some gene expression data for a variety of virulence genes and showing that the mucin glycans are able to downregulate these virulence factors where the monosaccharides have no effect. This is also true for aggregate. Aggregate size is smaller in the presence of MUC5AC glycans, but is not impacted by monosaccharides. And the same trend holds for a surface-associated biofilm model. And so what this paper really suggests is that changes to mucin glycosylation patterns during disease could alter not just the ability of bacteria to bind to these mucins, but also could change the physiology and the virulence profiles of these microbes. And so as we know, mucin glycosylation is different in CF. It's also different in response to a number of respiratory infections. And so I think this opens up some fascinating new experimental models and new questions that we need to ask and think about when we're thinking about how the respiratory environment could be impacting microbe function. And so concluding thoughts for this section is these new studies that were published in the recent year on finally some mechanisms underlying the well-known worse outcomes in Pseudomonas infections for females with CF. Also highlighted this resurgence in looking at metabolism in the host as a potential determinant or variable in infection outcomes. For anyone who's really interested in this idea of immunometabolism, Alice's team has also published a really nice study on Staph aureus small colony variants and the impact of glycolysis in the host in pathogenesis of these microbes. And so it's another great paper that I just didn't have time to talk about, but if you're interested, you should read this paper. And then finally, I just love this idea in this study looking at mucin glycosylation patterns and how they can shape bacterial physiology and virulence. And I think it's something we really need to think about moving forward in CF. And so finally, I'd like to talk quickly about some new and improved tools and models that might shape how we do CF microbiology. So the first paper I'd like to highlight is from Marvin Whiteley's team at Georgia Tech. I need to give a brief disclosure here that I am an author on this paper, but anyone who's heard Marvin talk about his work in the recent years would know that this is definitely the brainchild of Marvin's team, and we just contributed some RNA sequencing to help them flesh out some models they were studying. And so I'm really excited about this paper. And so what Marvin's team did is they developed this quantitative framework that took RNA sequencing data from a whole host of Pseudomonas infection models, as well as a large number of CF sputum samples with Pseudomonas aeruginosa. They looked at this RNA sequencing and they came up with this quantitative framework that allowed them to evaluate our Pseudomonas aeruginosa models to see how well the gene expression in those models replicates what's happening in CF sputum. And so when they ran all these models through, they ended up with two models that came out on top. One is the artificial sputum media that Marvin's lab developed, but many of us in CF use this media for studying Pseudomonas and other bacterial responses. The second model that showed really nice accuracy scores is the Airway epithelial cell co-culture model that was developed at Dartmouth and then I run in my lab as well. And so these two models are interesting in that they had the highest accuracy scores, but what was really interesting was both models underperformed, but in distinct categories. And so this raised the idea that we could use this quantitative framework to tell us where different models have different strengths. And so we can think about that as we're choosing which model we should be using to study our particular research question in our laboratories. We can also use this quantitative framework as a mechanism to improve our models and see where our models are deficient and where we might improve them to make them more closely replicate CF sputum moving forward. The last paper I'd like to highlight is a paper from Ryan Hunter's team at University of Minnesota, which is a really cool technique aiming to develop a new method to label translationally active subpopulations in CF lung microbiota. And so Ryan's team developed this labeling approach in response to frustrations in drawing mechanistic conclusions from microbiome studies in CF. And Ryan's team hypothesized that these issues with drawing mechanistic conclusions could be hampered by a variety and a variance in the metabolic activity of taxa in the microbiota. And so the way they got around to address this and actually look at specifically translationally active versus inactive taxa in the microbiota was developing BONCAT. And so BONCAT uses a non-canonical amino acid, and it's incorporated specifically in translationally active bacteria and allows you to then look in a complex sample and see which microbes are translationally active versus which are more dormant. And so when Ryan applied this to some sputum samples, I'm showing one from his paper here, you see with a variety of morphology of microbes, you also see a variety of BONCAT labeling here, suggesting there's great variability in different microbes and their translational activity. In Ryan's paper, they also showed great variability from sample to sample in different patients. And so that was also very interesting. Ryan's team also developed further the ability to use FACS to actually separate BONCAT labeled cells from unlabeled cells. So you can take your taxa plot, where typically in the original you would get just the taxa plot. Now you can know in that taxa plot which microbes, which taxa are metabolically active and which are more dormant. And so when he applies this, you can see in the red box I have here, some of these species that showed up in the original taxa plot are not translationally active, and some are very translationally active. And so this starts to give you some more information and more insight in trying to interpret taxa plots when you do 16S sequencing of your microbiome. And so I thought this was a fantastic study. It also gives us something to think about when we think about targeting antimicrobials and which microbes are actually active in sputum. And so this is another therapy that another model and new tool that I thought was really important to note. It was a fantastic study and gives us some new tools moving forward in CF microbiology. And so those are the papers I have. I thought there were a number of great other ones that came in as honorable mentions, but I only had 20 minutes. And so I hope you enjoyed this. I hope it was informative, and I'm happy to talk in the chat. Thank you.
Hi, this is Paul Planet, and I am going to be presenting influential papers in translational science in microbiology and infectious diseases. I'd like to thank the organizers for inviting me to give this talk and to highlight some really great work from other people. I have nothing to disclose. So first off, I'd like to tell you how I chose the papers. I'm going to be talking about 5 papers today. I decided that I would like to choose one paper from each category having to do with microbiology and cystic fibrosis, from the respiratory microbiome to the gut microbiome to the impact of antibiotics, single organisms, and then bacterial interactions. The other criteria I had for picking was that I wanted at least one aspect of each paper to really challenge some common conception. And we have a bunch of papers that do that today. And then I also wanted, because this is the translational portion, to have human subjects or specimens involved in all of them. First, to the microbiome. So the microbiome has been a rich source for publications over the past 10 to 15 years in the cystic fibrosis world, and there are many papers to choose from. And so I just wanted to highlight some of these ones that I'm not going to be talking about today, but are really worthwhile checking out, amongst others, by the way. The microbiome paper that I chose to talk about is one out of the group, out of Mike Surratt's group, entitled Culture-Enriched Metagenomic Sequencing Enables In-Depth Profiling of the Cystic Fibrosis Lung Microbiome. In this paper, they really tackle the major problems with current metagenomic techniques in CF. And these are that low biomass, although that's less of a problem in CF than in the normal respiratory microbiome. Also, the very high quantity of host DNA that needs to be sequenced in metagenomic techniques, and then the inability to get good assemblies. So in this paper, they advocate for a technique where they take sputum, this was mostly sputum in this paper, and then they culture on multiple different culture media and under aerobic and anaerobic conditions, and then go right then to sequence what they grow on those plates. This is done alongside direct sequencing from the sputum itself to gauge the relative abundance of the organisms that are there, because of course culture distorts those. And I'll come to the PLCA portion of this paper in a little bit. What this does is a really interesting thing. So if you culture in a lot of different media in different atmospheres, you can actually really enhance a number of taxonomic categories that you can isolate. And this is just a graph showing the comparison between the taxonomic categories found by direct sequencing and then compared to culture-enriched sequencing, where you can see a large increase. Of course, there is also a benefit to direct profiling, because you do pick up a few additional taxa from direct profiling. So really, these are things that are meshing in the middle. From this figure from the paper, you see that the kinds of things that are found on the culture only, that are found taxonomically on the culture only plates are really spread throughout the phylogenetic tree. And then there are a couple of smaller things that you pick up on direct sequencing. Of course, this kind of an approach to microbiome analysis requires a huge amount of labor, and then of course a lot of plating, and each one of those plates can be its own job to make. So one thing that they have that they suggest here is the plate coverage algorithm. They spent a lot of time developing this in this paper, which is the minimum number of culture enriched, which defines the minimum number of culture enriched plates necessary to capture the taxonomic diversity of a sample with culture enriched metagenomics. Basically, what this technique does is it uses 16S data from direct sequencing to then go in and figure out the number of plates that really need to be cultured. Of course, you will also pick up additional taxa because you can't know a priori what is going to grow from each sputum sample. But what it allows you to do is drastically cut down on the number of plates that you need to sequence, that you need to grow from, and then also sequence. So the take-home points from this paper are that the CF microbiome is largely culturable, and some of it can only be seen using culture, though there are some things like spirochetes that remain difficult to culture. And I think the major idea here is that combining culture and culture-independent techniques really increases the sensitivity for taxonomy, as well as, and this is part I didn't really talk about, for gene and functional detection. Moving on now from the lung microbiome to the gut microbiome and its impact on cystic fibrosis is a nice paper from Luke Hoffman's group called Fecal Dysbiosis in Infants with Cystic Fibrosis is Associated with Early Linear Growth Failure. So the rationale behind this paper is that infants with CF are known to have pancreatic insufficiency that results in malabsorption and poor growth. This can be compensated for, and has been shown to be compensated for, with enzyme replacement and nutritional intervention, at least for attaining weight by 1 year of age. But most of these infants still have poor linear growth in the first year of life. So earlier linear growth deficiency is associated with, has been shown to be associated with worse long-term respiratory function and survival. But really, the cause of this growth failure is not understood. So this is this paper really starts to get at the underlying mechanism of this. And it really asks the question, does the microbiome affect growth, that linear growth? So along these lines, components of the microbiome can impact endocrine function. We know this from animal models. And inflammation and malabsorption can lead to dysbiosis in the CF-GI tract. So putting this together, is it possible that the dysbiosis is actually leading to a difference in endocrine function? And if the answer is yes, this could be a modifiable target. So this study took 207 infants prospectively from the BONUS study and collected during the 3 to 12 month region fecal ages fecal samples. They did metagenomic sequencing, so sequencing all of the DNA there, not just 16S taxonomy. And then they compared that to age-matched healthy infants. The first finding is that the fecal microbiome is altered and delayed compared to healthy controls. So CF kids have more proteobacteria, less Bacteroidetes, and that and they attain diversity that is similar to normal late. And this was done in the paper using a whole bunch of different classifiers that is beyond the scope of this quick talk right here. But basically, the idea here is that the CF microbiome in these early is delayed and develops more slowly and has this predominance of proteobacteria. They then looked at, they split the CF group into normal length kids and low length. So those kids who were having linear growth that was below standard growth curves. And so even within CF, you see here that the low length CF patients have that increased proteobacteria compared to Bacteroidetes. The take-home model at the end of this paper is a really interesting idea. The idea here is that malabsorption and altered nutrient content actually lead to a difference in the makeup of the microbiome, which has low Bacteroidetes, and Bacteroidetes are well known to make the short-chain fatty acids butyrate and propionate. Those, in turn, have been shown in other studies to be involved in making IGF family growth factors or provoking IGF family growth factors to be released, and therefore the lack of these may lead to decreased linear growth. It's going to be really interesting to see how this hypothesis is borne out as the investigators tackle each one of these arrows. Next, also from the lab of Luke Hoffman and others, is a nice paper looking at the impact of tobramycin, inhaled tobramycin, on the microbiome. So tobramycin is the most commonly used antibiotic in CF. It's used as a maintenance therapy in patients that are otherwise at baseline. So this is an important point. These patients are otherwise healthy. They're not necessarily in exacerbation. It is well established in multiple studies to improve long-term outcomes. The problem is that the effects on Pseudomonas appear to diminish over time, and the effects on other significant CF pathogens also seem to diminish over time. So there's a general paradox. What is the tobramycin doing over time? So they decided to address this, looking at 30 patients with CF. Sputum was collected before, during, and after a month-long course of maintenance, a course of maintenance inhaled tobramycin. And they used traditional culture, quantitative PCR, and metagenomic sequencing to interrogate what was going on with the microbiome. So very quickly, when you look at basic culture, tobramycin impacts the microbiome in a relatively predictable way. There is in this sampling of some of the in some of the figures, there is a decrease in the overall amount of gram-negative organisms in week 1, and then this pretty much plateaus and then returns to normal levels after follow-up. In the multidimensional scaling plot at the bottom, you see that there appear to be there's a change in that centroid that goes from the baseline to the perturbed states when we're looking at tobramycin therapy. So that's a pretty predictable change, and maybe one we could have seen coming into this. But interestingly, when you look at the organisms that are involved in the change, the list of organisms that change most in when you treat with tobramycin are actually not the ones you would normally think. So these are organisms that are not on the top of our minds when we're thinking about treating for cystic fibrosis infections. And just to show this in another way, this is a list of organisms that is most changed this is in absolute abundance now, and you can see that there are very big differences between these organisms, but they are again not the organisms that we are most worried about when we're talking about treating. Then an unusual and unclear result from this was that there appeared, because they had metagenomics, they could look at lots of different functional properties inferred from the gene content of these samples, and they showed that there was not a huge amount of difference in function inferred from the metagenomics when you're on tobramycin or not on tobramycin. This is an interesting result because what it suggests is that there's some sort of redundancy in the gene content of the metagenomes that are there. It also could just mean that we're missing small but significant changes in the gene content. This is also, it's worthwhile mentioning this other paper from Hirali et al. Back in March 2019 that also showed sort of off-target effects for inhaled antibiotics. The next study that I'd like to talk about is a study by Stapleton et al. From clinical infectious diseases very recently over the summer, and where they wanted to look at the genomics of Pseudomonas aeruginosa in early infection in children with cystic fibrosis. So going into this study, it's worthwhile saying that most people believe that Pseudomonas aeruginosa infection is thought to be largely from environmental exposure, with the exception of some circulating clones that have been shown to circulate between people. But the large majority of it, especially early infection, seems to be from environmental exposure. And we have now entered the whole genome era, where prior molecular techniques of typing have become basically outdated in their because they are not able to get to the granular level that we can get to with whole genomes for establishing the sameness of strains. So when we want to talk about molecular clones, we want to be able to say that they're coming from the same, that they are the same organisms. So this group wanted to look at how much strain sharing there was, like how many people share the same organisms. They looked at 70 patients, 87 isolates of early pediatric Pseudomonas aeruginosa infections. They did whole genome sequences of that and really had an excellent control group. They compared it to chronic CF infection, over 300 isolates there, and they also did some environmental sampling, which I found really great, because they were able to get a handle on what environmental exposure might look like for these people. They also looked at sterile site infections and asymptomatic carriage. And they assessed possible transmission based on patient records when the patients were in the clinic, et cetera. When you talk about sameness in whole genomes, you have to figure out what you mean by the same. And what they were able to do in this study was to look at intrapatient variability of Pseudomonas strains by sequencing more than one strain from each person. And they found that sameness in their group using their methods was a cutoff, a very strict cutoff of 4 SNPs. It's worthwhile saying that this is a very strict cutoff, and this may have to do with the way that they processed and quality checked their sequences. But the long and the short of it is that they found that 41% of the early infection cohort had shared strains, and those were 14 different strains. That was a much higher number than we usually think of, and makes us rethink the epidemiology of Pseudomonas. One interesting point that they raised was that there were only a few epidemiological links, and you can read their paper for the specifics of this, but there were very clear and close relationships between these strains. And here's just one tree where they show the extreme closeness of these strains. Some of them are identical, being less, having 0 SNPs, but being from different people. I think one major thing to remember here, and this is something that has recently been an issue in the mycobacterial transmission literature, is that SNP cutoffs vary greatly from study to study and really depend on how you're making your SNP calls. So it's really important to be aware of them and make sure you know what the numbers mean. The last study I'd like to cover is from the Tony Fisher's group and Dominique Limoli's, and it's looking at co-infections with Staph aureus and Pseudomonas aeruginosa. This brings up the often shown figure you see in the bottom right hand here, where you see this inverse relationship between Staph aureus and Pseudomonas aeruginosa. The major point of this figure is that it is cross-sectional and does not represent a longitudinal sampling. And therefore, you cannot draw from this that there is an inverse relationship between Staph aureus and Pseudomonas. But Tony really wanted to see if in a longitudinal sample, you could pick out an inverse relationship between the two of them. So he has an excellent collection of many years of strains, and was able to select 134 children and adults from 2004 to 2017 with more than 10 cultures, observed over very long periods of time, and that had quantitative cultures for Staph aureus and Pseudomonas. They calculated the duration and the persistence of infection, both for Pseudomonas and for Staph aureus. And they were able to actually recreate the same cross-sectional pattern that you see in panel A here that you saw in the registry data. You can see that Pseudomonas dominates in the younger age groups, and Pseudomonas dominates in the older age groups. But when you look at any particular person, for instance, in a single subject here, longitudinally over time, there is a very much more complex picture with some with Pseudomonas sometimes dominating and Staph aureus sometimes dominating. Again, they also saw some evidence against direct competition. That is, when you plot Pseudomonas CFU versus Staph aureus CFU, you actually see a large increase in the, you see that both of them tend to increase together when in the same culture, so that there does not seem to be an inverse relationship, at least in quantitative CFU. One interesting result from this was that there does seem to be a difference in the duration and persistence of these different strains, with Pseudomonas aeruginosa being more durable and more persistent than Staph aureus, which may play into some of the things that we're seeing, some of the results we're seeing here. Overall, there was a net trend toward co-infection. Almost never saw a replacement of Staph aureus by Pseudomonas aeruginosa, which is very interesting because it goes against the idea that there's an inverse correlation there. So where does that leave us with the cross-sectional data? Well, there must be some reason why Pseudomonas in those older age groups dominates, and Staph aureus dominates in the younger age groups. And hopefully we will be able to figure that out in the coming years, especially as things change. Some factors are listed here, but really we don't know what the underlying cause of that pattern is. So to sum up the entire talk, I'd like to just take the major point from each one of these and how this might change our thinking. So in the first study, we have microbiome studies have come full circle to where culture is informing culture-independent studies, whereas before it was quite the reverse. In the second study, we have this really interesting finding that the microbiome may have a direct impact on linear growth by making a hypothesized short-chain fatty acids that go and impact growth factors. In the third study, we have the idea that tobramycin has its biggest impacts off-target on non-dominant organisms, and it will be really interesting to see if that is an important biological effect that contributes to the clinical benefits of tobramycin. In the fourth study, we saw a much higher rate of Pseudomonas aeruginosa strain sharing, and that was and that may change our ideas about what best practices are in epidemiology and in infection control. And then finally, despite the common assumption that Staph aureus and Pseudomonas compete, Pseudomonas did not seem to outcompete and eliminate Staph aureus in a longitudinal cohort. Thank you very much.
Good day, everyone. The new normal, a phrase we've all heard so many times in the pandemic. I'd like to see how we can apply that to pharmacokinetics and clinical application of antimicrobials in the 21st century for our patients with CF. The picture on the lower left is the UK Med Center, where both the children's and the adult hospital is, where our centers are. And hopefully a beautiful day that's in where you are today as well. Disclosures with Vertex Pharmaceuticals, no direct bearing on today's talk. So let's think about the historical evidence of pharmacokinetic differences of anti-infectives in patients with CF. Aminoglycosides, beta-lactams, anti-staphylococcal penicillins, and maybe things have changed over that period of time. And let's look at some new evidence of some of the new anti-infectives and some of the PK and PD work that we have. And then how does that translate into our new prototypical patient with CF and what clinical impact it might have. Goals of treatment have tried to pretty much been the same to prevent and control lung infections, especially Pseudomonas and MRSA. We've used aminoglycoside dual therapy, typically with beta-lactams, carbapenems, etc., monobactams, and vancomycin, especially with patients co-infected with MRSA, which require careful monitoring. And so we have to tailor the doses of antibiotics to maximize therapy. We certainly don't underdose, nor do we want to overdose, so that we can minimize short-term toxicity as well as long-term or cumulative toxicity. So there's some questions that sort of arise when you think about what we might think about today, you know? What are the right doses for these new anti-pseudomonal compounds? What data do we have for that? On these beta-lactams, which we'll kind of focus on today, should we use prolonged infusions, you know? I've always used higher doses, but maybe some of this other data might help me better understand how we should approach it. And then sort of the unknown is, how do modulators, or at least effective modulators and therapies changing, how might that change even the pharmacokinetics of these drugs? What's new in CF chronic management in the last 10 to 15 years? Well, from an antibiotic standpoint, chronic azithromycin, using more and more oral anti-pseudomonal and anti-staphylococcal antibiotics, as well as, the reliance on now highly effective modulators and how that might have an influence. These are still somewhat, we're still learning the implications of that. So probably a paper that I thought really got things started with Bolita and group in Clinical Pharmacokinetics about a year ago, published. They looked at 4 decades of CF data relative to PK and beta-lactams, even anti-staphylococcal penicillins like diclox and gloxycillin, cephalosporins, carbapenems. Take all those together. Let's compare them compared to healthy volunteers by publication age, adjusted for age and weight, using called allometric scaling. And that's a technique that people are using now to sort of make those pharmacokinetic and at least pharmacokinetic changes. And they also found that in looking at this, the protein binding effect, especially for clox and diclox, dramatically diminished. And other clearances were much closer to healthy volunteers. So maybe 3 buckets or 3 groups. CF patients as group 1 early on, much younger, much lower body weight, body surface area, and they were much sicker. Group 2, a lot less of a difference in age, but still a pretty substantial change in body weight and lean body weight. And group 3, this group, when you look at some of this modeling, came within 10% or so of matched controls. And really, the PK modeling accounted for these differences. We would call those pretty much similar. And it's really this 40 years of data is changing, and our patients are healthier, they're living longer. So changes are really in the offing till we need to reconsider some of these approaches. I would say the takeaway point from this paper, and we'll go much in more detail, is that cephalosporins really should be targeted by targeted concentrations. What's that MIC? Knowing that we don't have that good correlation in CF, at least as a target, and really may not be an intrinsic factor of the clearance of the drug at all, or at least minimally. And really none of these were done with highly effective modulators, and that might have some role as well. This was that model assumption, looking at half-life of the dotted line, clearance and volume distribution, adjusting for body surface or body weight compared to the 70 kilo patient, okay? And this was the model that they used and really has been used in other areas, but they proposed this in CF and actually maybe could use lean body weight even in CF. But they came up with these 3 groups. The cartoons are directly from the paper, and I think it makes perfect sense. In the 70s, 80s, they were much younger CF patients than healthy volunteers, and they were much healthier as healthy volunteers compared to our CF patients who had very small body weights. They were ill and clinically not doing as well. And so you'd expect some differences in pharmacokinetics. In group 2, which is 30 to 40 years ago, that more age match for sure, but they're still less healthy, lower body weight, body surface area. So those kinds of differences were quite different. And I applaud the authors for making this rather cartoon-like, but easy to follow. And groups 3 were the groups more recent, but still up to 20 years ago, much more age match, healthier CF patients compared much closer healthy volunteers. And their data shows, and this is a summary of that, pharmacokinetics markedly increased back in the 70s, some increased still in the 80s and 90s. And now with some of these new techniques, really, they can account for up to they can only not account for up to 10 to 20% of clearance. And with protein binding, especially the anti-staphylococcal penicillins, that protein binding really diminished to more modest 20 to 30% compared to these very large differences that were marked back in the 70s and 80s. So if you think about it, those patients were dramatically different in the 70s, 80s. They were different for most of our patients in the 80s and 90s, but now our patients are healthier. They're closer to body, good BMIs, and really looking at that as a goal in lung function. So how does this impact? Well, let's look at the newer agents, the newer beta-lactams, ceftazidime, avibactam, and ceftolozane tazobactam. There were a pair of papers that came out. First one was the PK target attainment. Now this is looking at target attainment. Can we get the MICs that we would want in case they chose 16, all right? Gave adult CF patients 2.5 grams every 8 hours, pretty much as labeled, but they gave it over a 2-hour infusion to try and get that optimal beta-lactam performance. They use intense PK sampling and these Monte Carlo simulations, which allows you to do so much with so little data. And they really found that this demonstrated the clearance was similar, the PK was similar to labeling for adults. But now we're going to use that caveat, the direct comparison of clinical outcomes and traditional MICs and CF is at best questionable, but I think it's something we go on now to at least give us an inclination of how to treat. So this well-accepted model accounted for a great deal of this change, and really there wasn't much change. And the group, Microbial, looked at the same thing with ceftolozane tazobactam and found similar results. Modest changes, but not dramatic, okay? So again, coming back to those same things, determined by target concentration, and none of these studies were done with highly effective therapy, okay? So these are the actual work that the group did, Benzema. They looked at probability of target attainment, so with low MICs, 16 I wouldn't consider low, but you can see pretty much they can hit the target they want. When you hit 32, then you dramatically lose your target attainment, which means about 80% of patients, the CRP, would be a good respond. They looked at, this is just another way of looking at probability, looking at MICs, 8, 16, and 32, and you can see you get 32, you probably plateau out at about 35%, okay? And they use 50% as target attainment. Some people use 40, 50, 60%, okay? So ceftolozane tazobactam, this is a Montague's group. They saw similar kinds of changes, but they saw similar clearances, but a smaller volume of distribution. Again, they use that same kind of technique, but now they chose around 40%, 39%, 60%, and 100% time above the MIC. And so a lot of times, at least traditionally, people use 40 to 60, but people might even argue with CF patients and their multi-drug resistant organisms, could we even shoot for a target of 100? You can see that is possible for MICs, okay? So again, another 2 studies that showed that the population pharmacokinetics was modestly different, but not dramatically, but probably looking at target attainment about how aggressive one would be in dosing and how you would get it. So what other PK considerations do we have? Well, we have seen increased clearances of ceftaroline, so this is a little different than just talking about the other 2 cephalosporins. The method of administration does make a difference, at least in target attainment, and changes in clearance over time during treatment. And that's something we've noticed, and clinically over my 38 years in taking care of CF patients with the CF Center. And to talk a little bit about this. So Thompson and their group back in 2016 looked at these Monte Carlo simulations of 5 anti-pseudomonal beta-lactams and said, okay, here's the doses. This is how we typically give patients drug, okay? If we give it as a bolus to reach this PTA of 90%, so we used a little higher watermark, not 100, but 90%, could we reach those target attainments based on a bolus infusion of 30 minutes, a prolonged infusion of 3 hours, or literally doing a continuous infusion, okay? And you can see that just by going from bolus to continuous infusion, you see some pretty marked increases, cefepime from 32 to 66. Now they actually use isolates, CF isolates from the center, CF Center there to look at their MICs and stuff. So these weren't just stock Pseudomonas, these were Pseudomonas from CF patients, both adult and pediatric, okay? Ceftazidime 56, 75. So you can see a pretty dramatic increase, by using just either prolonged infusion or continuous infusion. I think that's a takeaway I hope you can think through as you take care of patients. Elizabeth Audrey and her group looked at ceftaroline a few years ago, looking at 40, 60, 80, and 100% of the dosing interval, trying to hit the MICs, okay? And you can see that D is the one I really want to highlight. That's your prolonged infusion of 3 hours. And you can see that target attainment can go way out here for higher MICs. You think of, for ceftaroline, 1 or 2 micrograms per mil. And you can see some pretty good responses out there. And obviously, for more rapid infusions, the times go away. Now, 40, 60, 80, 100%. I wish I could tell you which is best for CF patients. Clinically, certainly 40 to 60 is more the traditional one in some of the other models. But I think given what we know about CF and stuff, one might argue, can we actually get 80% or 100%? Their group also looked at PK and saw that really to simulate these kinds, they really need to give every 8-hour dosing of ceftaroline in adults versus every 12 as labeled because of changes in clearance. So this was, I believe, 8 or 10 patients. It was a prototype study. And I think showed that at least clinically, that's what we do is give it every 8 hours with this data as a background. Now, the group in Utah looked at, hey, what happens during therapy between days 2 and day 7 of a probably traditionally 13, 14-day exacerbation. They saw between day 2 and day 7 a pretty significant increase in clearance, which means perhaps you might need more dose or higher dose. And with aminoglycosides in the blue, you saw a diminution of 10%. And we know these drugs probably accumulate, maybe changes in clearance and toxicity, et cetera. So one might project how is that out to day 10, day 14 as we traditionally treat our patients. I think it requires careful monitoring. I think that's the purpose of their paper, but also to point out the beta-lactam clearance. And we've actually measured it in select patients and saw a similar sort of effect. Now, we don't have as much data as Utah, and this is well-published data to show those kinds of changes. But again, not only changes in clearance, but clearance over time. Okay? So what is the impact? Well, I would suggest that prolonged infusions of beta-lactams should help us with targeted success in our patients. I would consider making this a standard to use in practice. It's not saying what dose is different, it's not saying how you pick the drug, but if you're going to use it, it makes sense to sort of give the maximum pharmacodynamic approach to the drug, okay? And that much of this is based on that targeted concentration. What's the MIC? How's the patient doing? That kind of stuff versus absolute changes in pharmacokinetics. We didn't talk about it, but I just want to mention that vancomycin aminoglycosides, tread carefully, especially using in combination. We tend to go more with AUC monitoring, especially with vancomycin. We're looking at exploring that in aminoglycosides as well. I know some other centers are, but I think really helps us better think about how we can optimize our patients, okay? Given all this, I just think it's a necessity to have a pharmacist on the team who can help with all this PK work, and they need to be skilled and understand that. And I appreciate the fact that the Cystic Fibrosis Foundation has helped coordinate and help co-fund some additional pharmacists for CF centers, and I would consider them not only a recommended team member, but hopefully someday a required team member. And in many centers, I think the center directors and team feel they're required already. So I'm just putting a plug in, and I hope your pharmacists are working closely with you. Because one of the roles is the pharmacokinetic and therapeutic drug monitoring, because I think we're getting more sophisticated. We're looking at target concentrations, AUC, decreasing toxicity, changes in clearance, and all those things really need to be codified inter-center. So the one missing link, or the one additional link, is now we have these class mutations, we have our favorite 508del, and these different class mutations. As we now use effective modulator therapy, what might that mean? What might that mean on how often we have to treat? One might argue less, and the future of that. And is this really group 4? this group of patients who are going to be on modulators, whether it's the ones who currently have or even better ones or combinations, also maybe gene therapy, what will group 4 look like? They may be very close to normal because of their wonderful nutrition and lung function, at least initially. So in closing, these are some caveats that go a little beyond just the beta-lactams. I think with fewer exacerbations, we can aggressively target 10 to 12 milligrams per kilo of tobramycin once a day and adjust according to pharmacokinetic parameters. I think transient increases in serum creatinine are reversible if carefully monitored, and the monitoring is important. And it may have a lot to do with BMI and health status. Remember, fluid status is important on aminoglycosides. There's a publication in Press looking at cumulative toxicity and really was very unimpressive long-term, even doses up to 6 and 8 grams per kilo cumulatively, okay? Dr. Sed's work will be published soon. Less exacerbations in the era of modulators probably will decrease our ability to worry about long-term toxicity. So maybe we need to be aggressive when we have to treat. And I think the AUC approach to vancomycin and perhaps even aminoglycosides might be the way to go. But I think for vancomycin, there's certainly data in the non-CF patients that AUC is desirable. And I think we've started to use it in our CF patients as well. And I think we're seeing less toxicity with hopefully similar outcomes. Thank you very much for your time, and I look forward for interactions online.
Part of the symposium, Year in Review, Influential Papers and Topics in IDM Microbiology, and I'm going to be discussing the clinical topic. My name's Elizabeth Tellis, and I'm the CF Clinic Director in Toronto, Canada. I have a few disclosures to make related to this presentation, in that I have received funding for research studies from Novartis and Gilead in the years listed here. Now, today I've chosen to present 3 papers within the same theme. And the reason for this is I'd like to address an emerging relationship between azithromycin and tobramycin in the CF population. These 3 papers were all published within the past 15 months. Now, why I decided to choose these papers is that these drugs, azithromycin and tobramycin, are an important part of the standard care of patients with CF. Azithromycin was shown in phase 3 trials to reduce pulmonary exacerbations in patients with CF, and now 64% of patients with CF in the US population are on this therapy. Tobramycin has also been shown to be standard care for both treatments of pulmonary exacerbations as well as chronic therapy in the inhaled route for people with Pseudomonas. And the prevalence of inhaled tobramycin in the US CF population is 70%. And so I think it's important for clinicians to be aware of emerging evidence to suggest a negative interaction between these 2 important therapies. Now, the studies that I've chosen reflect the use of tobramycin, both in the long-term chronic Pseudomonas use of inhaled tobramycin, as well as tobramycin when used by intravenous route to treat pulmonary exacerbations. And this has been shown using the registry data as well as single center studies in both the pediatric and adult population. Now, there is some biological plausibility regarding the relationship of tobramycin and azithromycin. And there's in vitro evidence showing antagonism. And this may be because azithromycin may reduce the ability of tobramycin to kill Pseudomonas through upregulation of the drug efflux pump, so that Pseudomonas gets better at getting rid of tobramycin in patients with azithromycin. So there is a kind of biological in vitro reason why there may be an antagonistic relationship. So to start off with, I'm going to talk about azithromycin in conjunction with the use of inhaled tobramycin in the chronic management of CF. And this first study was done looking at the American CF Patient Registry. It's a retrospective cohort analysis. And they were looking at 4 different groups. Those patients who were chronically infected with Pseudomonas and those patients who weren't. And in the patients who were chronically infected with Pseudomonas, those people on inhaled tobramycin and those people on inhaled aztreonam or ASLI. So there's the 4 groups looking at the use of azithromycin versus no azithromycin in these populations. The outcomes of interest are looking at the rate of decline in lung function over the 3 years of the study. In addition, looking at pulmonary exacerbations that required IV antibiotics, looking at the time to the first exacerbation or the rate ratio of pulmonary exacerbations. Now, of course, these outcome measures, rate of decline of FEV1 and pulmonary exacerbations, are impacted by many different factors. And thus, the populations have to be matched based on variables that are known to affect both the rate of decline of FEV1 and the frequency of exacerbations. And on the right-hand side of your screen, there's a list of the 14 variables that were used in the propensity score to match the patients in this study. Now, the first group studied were patients who were either chronically infected with Pseudomonas or not infected with Pseudomonas. And when they looked at the registry data and they looked at narrowing down the population to make sure that there was enough years of follow-up and there was enough time on azithromycin, and applying the exclusion criteria, making sure that this population had good data to be very sure of the Pseudomonas status, present or not, and then the propensity score matching, they ended up with about 1,500 patients, equally divided into those patients who had Pseudomonas, chronic Pseudomonas, and those who didn't. Now, in terms of the results, this figure shows both the results of patients who are not infected with Pseudomonas and those with Pseudomonas. So we'll start on the left-hand side, those patients who are not infected with Pseudomonas. And the red indicates patients on azithromycin, and the black indicates patients who are not on azithromycin. And this slide is looking at rate of decline of FEV1 over the 3 years of the study. And you can see in this initial slide, the red line and the black line are lying on top of each other, meaning there is no difference in the rate of decline in lung function in patients who are not chronically infected with Pseudomonas who are either on azithromycin or not. So no difference. However, the second column, which is part B, or on the right-hand side of the slide, shows those people chronically infected with Pseudomonas. And in this case, there is a difference. Patients who are on azithromycin have a slower rate of decline of lung function, whether you measure it looking at all lung function measurements over this time course, or just looking at the best FEV1 in the year. And in fact, there is a 37% slower decline in the slope of the yearly maximum FEV1 in patients on azithromycin. So this is showing that in a similar positive fashion to the phase 3 trials that showed a benefit of azithromycin, this registry study is showing a benefit in long-term azithromycin, not just looking at an acute improvement in lung function. Now this is looking at the slowing of the rate of decline in lung function and showing that in people with Pseudomonas, this azithromycin is helpful. Now, what about looking at the people with Pseudomonas and saying, well, what happens if you're on tobramycin versus if you're on aztreonam? And so here they looked at the population of patients who were either on inhaled tobramycin or aztreonam, once again, applying the criteria of appropriate follow-up and being very sure of their therapy, and applying the exclusion criteria and the propensity score matching. And there they found that at the end of the day, that of the almost 5,000 patients, 4,000 of them were on inhaled tobramycin, and so 80% on inhaled tobramycin, and the remaining 20% on inhaled aztreonam. And the patients on the tobramycin were younger with better baseline FEV1. And that makes sense because tobramycin is sort of the #1 choice for patients with chronic Pseudomonas and tends to be the first drug started in that patient population, whereas aztreonam tends to be sort of what is done after perhaps people have not done so well on tobramycin, so older and less well patients. Now here in this table, they're showing the slope of decline in FEV1, looking at all the data versus the best data. And so I'm looking at the bottom part, the circled numbers, which is looking at the % predicted FEV1 decline per year using the best FEV1 in the year, and comparing the difference in people who were using azithromycin or not using azithromycin. And when you look at the patients on inhaled aztreonam, you see the same result as the overall group, that aztreonam use, those people on inhaled aztreonam and on azithromycin had a slower rate of decline in FEV1 % predicted per year. But if you look at the column with tobramycin use, there was not this decline in rate of decline of FEV1. So that benefit of the azithromycin in the Pseudomonas patients was not seen in Pseudomonas patients who were on inhaled tobramycin, but it was in those on aztreonam. So in conclusion, patients with CF who are chronically infected with Pseudomonas have a slower rate of decline in FEV1 after they start on azithromycin compared to patients who are not on azithromycin. And that was seen in patients with Pseudomonas, but not in the population of patients who did not have chronic Pseudomonas. However, this finding was not seen in patients with chronic Pseudomonas who were on inhaled tobramycin. And there, in that case, the benefit of the azithromycin was not seen. However, in patients who were on a different inhaled antibiotic, inhaled aztreonam, did show the beneficial effect of azithromycin, which is associated with a slower rate of decline. So now I'd like to move to looking at azithromycin in the context of treatment of acute pulmonary exacerbations with IV tobramycin. And the first study will be looking at a single center study in a large American CF center looking after adults. And there they reviewed the pulmonary exacerbations in their center that required hospitalization and IV antibiotics, and looked at patients who received IV tobramycin or IV colistin as part of their treatment of exacerbations. And then they looked to see if patients were either on chronic azithromycin therapy or not. The outcomes of interest were related to the recovery in FEV1 at the end of the exacerbation, which is obviously a very clinically important outcome for treatment of exacerbations. And so they looked at it in different ways. The ratio of the lung function at discharge versus baseline, the absolute recovery from start to end of therapy, and the proportion of people that recover to within 90% of their baseline or within 100% of baseline. Now, there's some challenges with looking at the analysis of azithromycin and FEV1 response in pulmonary exacerbations. And that's because baseline FEV1 is a factor on the causal pathway, meaning that it's a co-founder. So baseline FEV1 actually influences the likelihood of you being on azithromycin. So if you have lower lung function, you're more likely to be on azithromycin. But baseline FEV1 also influences your likelihood of responding to treatment of a pulmonary exacerbation. And so you have, you've got FEV1 working as a co-founder. But it's also a mediator of this interaction as well, because baseline FEV1 is influenced by azithromycin use. So people on azithromycin may have a higher FEV1, which impacts your response to pulmonary exacerbation. So when you have factors that are both confounders and mediators, there needs to be adjustment in the statistical analysis in order to come to correct conclusions. And the standard multivariate regression model is not capable of doing this, as it may produce biased estimates when it's adjusting for variables that are along the causal pathway. And so the authors needed to use different models, such as the marginal structural modeling, to be able to take into account factors that are both confounders and mediators. And so in this study, these different models were used in order to adjust for these time-varying confounders, such as an impact on the outcome measure. Now, the results of this study are looking at the patients who received IV tobramycin and the patients who received IV colistin, and looking at their absolute and change in FEV1 during the IV therapy, comparing those people on azithromycin with those people not. And if you look at the top part of the table and the people on tobramycin, that people who were treated with IV tobramycin and were on azithromycin had a lower recovery of their FEV1 than people not on azithromycin. So it's sort of saying, if you happened to be on azithromycin, you got IV tobramycin, your FEV1 did not increase as much at the end of your exacerbation. So the azithromycin, patients on azithromycin did more poorly. But this finding wasn't found in the patients treated with their exacerbations with colistin. They had a recovery of their FEV1 that was 2% greater if they were on azithromycin than if they weren't on azithromycin. So once again, showing that there seems to be an antagonistic relationship between azithromycin and tobramycin, with those patients being treated with tobramycin and azithromycin not getting the same kind of response that you would expect, in this case, to recovery in lung function after treatment of an exacerbation. Now, what about in a pediatric population? So this is also a single center study. This is a study from a registry in Canada, looking at a large CF center, and looking at patients who had chronic pseudomonas, who had pulmonary exacerbations, and they were treated with IV tobramycin, and the subjects were either on azithromycin or not. And once again, very similar outcomes: recovery of FEV1 from start to end of therapy, both relative and absolute, as well as the percentage of people who recover to within 90% of their baseline. And similarly, using the same statistical modeling to allow for correct for variables that were both confounders and mediators. And in a similar way, they found that patients who were on IV tobramycin and azithromycin did not have as good a response in their lung function after treatment of exacerbation. This study showed that there was a statistically significant relative improvement in FEV1, and that the patients who were on azithromycin had their improvement in FEV1 was 9.5% lower than in people who were not on azithromycin, although there was no statistically significant difference in the absolute response of FEV1. Now, there's some late-breaking science in this as well. And this is a publication that came out about 6 weeks ago electronically. And this is looking at the effect of use of azithromycin and tobramycin in treatment of pulmonary exacerbations in children. And this is, again, using the American CF Foundation patient registry, looking at children hospitalized for treatment of exacerbation that were treated with IV tobramycin, and looking at patients who were receiving azithromycin versus those who were not. Same outcomes: recovery of FEV1 in terms of from beginning to end of treatment, but an additional new outcome, which was the time to the next pulmonary exacerbation. Similarly, the statistical models used allowed for this correction of variables along the causal pathway. And the results showed that when they analyzed the registry for children who were treated with IV tobramycin for pulmonary exacerbation, they found that there were 5,000 exacerbations within this time frame. About half of them were in children that were on azithromycin at the time of the exacerbation. Half were not on azithromycin at the time of the exacerbation. And the results of this study showed that the patients on azithromycin who were treated with IV tobramycin had a lower absolute improvement in FEV1, had a reduced chance of returning to at least 90% of baseline FEV1, and in addition, had a shorter time to the next exacerbation requiring IV antibiotics. And this was comparing to patients who were on IV tobramycin, but they weren't on azithromycin. So in conclusion, these papers, the 3 initial papers and then the late-breaking paper, show that azithromycin and tobramycin seem to have an antagonistic relationship. And this is seen for the use of tobramycin as chronic inhaled therapy, as well as tobramycin used to treat pulmonary exacerbations. And what that means from a practical point of view is people on azithromycin who are also on tobramycin fail to get the benefit in the reduction in the rate of decline in lung function that is seen with azithromycin. And patients on azithromycin who were treated with IV tobramycin have a reduction in their FEV1 response at the end of exacerbations, and they have a shorter time until their next exacerbations. And if you look at patients that are treated with non-aminoglycoside antibiotics, be it inhaled as Trianam for the chronic treatment of Pseudomonas or IV colistin for the acute treatment of exacerbations, you don't see that antagonistic relationship. So these are all studies looking at registries or single centers. This is not, this is a placebo-controlled trial, and this is not prospectively collected data. And so we await with interest the results of the TEACH trial, which was a randomized placebo-controlled double-blind study of looking at patients with Pseudomonas on inhaled tobramycin who were randomly prescribed or given azithromycin versus placebo and looking at their outcomes in terms of lung function. And this study has enrolled, but we're still awaiting the results. And I think that the message here is an important message and something that we're probably going to see more of in the future. And that is, as we evaluate therapies for CF and we test a drug in a phase 3 study and show a benefit, which happened with inhaled tobramycin in people with Pseudomonas. And then we look at another drug, azithromycin, in a phase 3 trial where we see a benefit. And now we add the therapies together. We're discovering that 2 positive results added together may not make a super positive result. In fact, it may result in a negative effect. And this may be happening with other therapies or will be happening with other therapies in the future. And you can see that it's difficult for us to pick up these potential interactions and that the first hint that this is happening is from looking at the registry studies. And so we're left in a situation at the moment that we have some evidence to suggest this negative interaction. We're awaiting the results of a trial that might help us define the extent of this problem. And we're left with the thought that this may be happening with other aspects in our therapy for people with CF. So thank you very much for listening to the presentation of these 3 clinical papers. Thank you.